about molecular epigenetics. Now, if there are something that are not clear, please ask me. Also, I'm speaking in English, and I know it's not my language, it's not your language, so please, if things are not clear, please do ask me. Okay, so first of all, I want to thank Marian Lamb, with whom I did all this work, and Anna Zeligovsky, whose pictures you will see here, also illustrated our book. And I also, of course, want to thank all my students and my colleagues, and to thank you for being here, and the organizers for inviting me. It's always a great pleasure to be in Italy. Now, I want to start from what the challenge is. What is it that we want to extend, and why do we want to extend it? Now, the modern synthesis of evolution, on which I was brought up, and I think most of you were to some extent brought up, I don't know exactly how the textbook looked these days, but the modern synthesis, the way that we're thinking about evolution in the 20th century, in the beginning even of the 21st, was based on a bringing together of different fields of biology and of excluding certain things from evolutionary thinking. So it was both including things and excluding things. And there were, and this synthesis happened sort of between the late 1930s, and it sort of crystallized really around 1960s, but it dominated evolutionary thinking for about 60 years, of course, with variations. Everything always varies, and there were always extensions of the synthesis and so on. There was a group of American biologists that were the architects of the synthesis. One of them was Ernst Mayr. And Ernst Mayr was not only a biologist, he died recently at the ripe age of 102. And he was not only a biologist, but he was also a person who wrote quite a lot about conceptual issues in biology, philosophy of biology, and the history of biology, the history as he saw it. You know, there is a very well-known quote from Churchill, or attributed to Churchill, saying, history will remember me because I will write it. So, anyway, Ernst Mayr was one of the people who really were putting together this new synthesis, which there was a very famous meeting in 1947. I will show you a picture in 1 minute. What he said there, it was in these years, 1936 to 1947, that biologists of the most diverse subdivisions of evolutionary biology and from various countries accepted two major conclusions. One, that evolution is gradual, being explicatory in terms of small genetic changes and recombination, and in terms of the ordering of this genetic variation by natural selection. And two, by introducing the population concept, by considering species as reproductively isolated aggregates of populations, and by analyzing the effects of ecological factors. By the way, I have no arguments with these things. There's obviously, all this is okay, as far as I'm concerned. And he said, what the synthesis required, it required that the naturalists abandon their belief in soft inheritance, and that the experimentalists give up typological thinking and be willing to incorporate the origin of diversity in their research program. It led to a decline in the concept of mutation pressure and its replacement by a heightened confidence in the powers of natural selection, combined with a new realization of the immensity of genetic variation in natural populations. Now, what is very interesting is not only what was included in the modern synthesis, but what is very, very important, no less important, is what was excluded from it. And it is with what is excluded that I have an argument. So let us see just for a second. This is the famous 1947 meeting. You may see something slightly odd here. It's all, there's no one inactive X chromosome in this audience. The women were probably making the tea. But these are the architects of the modern synthesis. Now, I want to go with you a little bit, not in great detail, but I want to go maybe the gods are against it. Thank you. Right, so what are the assumptions of the modern synthesis and what do we argue about? We are arguing about the things in red. This is all not acceptable, I think. The things in blue, in black are okay. So what they said was heredity occurs through the transmission of germline genes. Hereditary variation is equated with variation in DNA-based sequence. What they said, there are no inherited non-DNA variations that cannot be explained in terms of DNA variation. If you see something that is non-DNA inheritance, in the end, it's okay. You can see a change in DNA-based sequence which can explain this. So any difference that makes a difference in heredity, in the end, can be explained at the DNA level. This was an assumption. 2. Hereditary variation is the consequence of 1. The combination of pre-existing alleles that are generated by the sexual process, and 2. New variation mutations that are a result of accidental changes in the DNA. Of course it's true. That's part of what we know about how variation is made. No, hereditary variation is not affected by the developmental history of the individual. There is no soft inheritance. Soft inheritance is inheritance of things that were induced during development and then passed on to the next generation. 3. Hereditary variation has small effects and evolution is typically gradual. Through the selection of individuals with phenotypes that make them slightly more adapted to their environment than other individuals are, some alleles become more numerous in the population. Of course, some evolution is gradual, some evolution goes exactly like this is described, no problems with this as part of the story, but there is also a stronger negative assumption, with very few exceptions. The exceptions were plants, polyhybridization, but they really didn't like it. Macroevolution is continuous with microevolution and does not require any extra consideration beyond those operating during microevolution. In this lecture, I will be particularly talking about these assumptions and about the exclusion, these 3 exclusions, but there is more to it. They also said the following, that the ultimate unit of selection is the gene. Although genes interact and interactions are often nonlinear, the additive fitness effects of single gene drive evolution by natural selection. Now there's again no question that additive interactions, that there are additive interactions affecting fitness, there's no question about it, but it is very clear that for them, that the genetic developmental network and the phenotype it generates is not heritable and cannot be a unit of evolution. Okay, so, and I will not go, there's also a strong argument again about the importance of physical constraints in evolution. What is their role in shaping evolution as we know it? Something, by the way, that Stephen Jay Gould was talking quite a lot about. He was talking about developmental constraints, and also he was talking about physical constraints, about the kind of thing that Darcy Thompson was talking about. So the role of physical constraints in the modern synthesis is not very great. It's as if the sky's the limit as far as variation is concerned. And the shaping of and the constraints and the affordances, the type of biological materials we have and so on, is not very important. We can explain everything with variation and selection. We don't have too much order for free, or great constraints of order. Another point is that group selection or community selection is not very important in the modern synthesis. There is a strong, at first it's not so clear. In the 1947 meeting, this was not a very big issue, but it became a very big issue in the 1960s. And the thing is that, and the other thing is that when we're looking at the pattern of evolution according to the modern synthesis is tree-like. Because we must remember, Ernst Mayr was a zoologist, and zoologists don't see very, or did not see, or did not recognize it, did not acknowledge the reticulate pattern of evolution that we know exists very much in plants, in fungi, in microorganisms. So we have an argument with all these points, not with the positive things they say about evolution. This is part of the evolutionary story, but the negatives. All the negatives are today challenged, all of them. Now, this is Dobzhansky, one of the fathers and the architects, a great geneticist of the modern synthesis, responsible for genetic or for the population field population genetic part and genetics. He came from Russia and brought from there a great tradition of field population genetics. And he said something that, you know, Dawkins could have said it. And this is what he said. Heredity in the last analysis, this is what he was specializing on, is self-reproduction. The units of heredity and hence of self-reproduction are corpuscles, little bits of macromolecular dimensions called genes. The chief, if not the only function of every gene, is to build a copy of itself out of the food materials. The organism, in a sense, is a byproduct of this process of self-synthesis. So this was said in 1958. It's quite early, and it gives a very strong sense of how people, very, very important and very influential people, were thinking about the hereditary aspect of evolution. They were very much concentrated on genes, not on phenotypes, and they believed that the only input that is important for heredity, for hereditary transmission, of course not the only input into development, but the only input that is important for heredity is genetic. Genetic, at that time, is already DNA. Now, and the picture that they have is this. We have genes. The genes, there's all kinds of things happen during embryogenesis. We have gene expression. The soma is formed. But the transmission from one generation to the next is only through the germline. And the germline is genes, DNA. This is the view. Now, the other person who was very influential is Ernst Mayr. As I said, he died at the ripe age of 102. And this is what he said. The crucial significance of the synthesis, then, is the fusion of widely divergent conceptual frameworks of experimentalists and naturalists into a singular. And I think this was really very important. He's right about this. And he said, in a symposium of Princeton in 1947, representative of the most diverse branches of biology, including ecology, came together and declared their acceptance of this new synthesized integrated evolutionary theory. Only the developmental biologists stayed outside, he said, insisting that the Darwinian theory was unable to provide an explanation of the evolutionary phenomena encountered by them. And indeed, it was recognized by Mayr and by other people that the one thing that was not really incorporated fully into the synthesis was developmental biology. And this was not because people didn't write about it. Because Waddington wrote a series of books from 1942 onwards that were doing exactly this, putting development into the synthesis. But they did not become part of the canon. They were not part of what people recognized as this is what evolutionary biology was about. There are always people who are outside at the periphery who are doing extremely important work, but usually 20 years after they die, people recognize this. Now, the other thing that Mayr was talking about, the importance of genetics, of what we know about heredity for what we understand about evolution. And he said it many times, it was perhaps the greatest contribution of the young science of genetics to show that soft inheritance does not exist. There is no such thing as what we can call Lamarckian inheritance. There is no inheritance of variation that are acquired during development and can be passed on. It just does not exist. And for him, it was very important, because what he was trying to do was to unify biology. And he wanted all these very different points of view about the role of selection and about this and about that and and there were neo-Lamarckians and there were people who believed in orthogenesis and there were all kinds of people. He wanted them all to unify around a set of organizing ideas. And so he had to exclude a lot of stuff from the synthesis, and he did. And he told us in a very dogmatic way, because he was a very dogmatic man, that this is how the world looks. So what we have is an evolutionary synthesis where a lot of things go in, and it's very important that they go in, and there is one little thing outside there. So It included systematics, comparative animal morphology, not so much by the way, but some of it, botany, also selected bits through Stebbins. But if you actually read what Stebbins wrote, he was the biologist of this synthesis, he didn't fit very well. By the way, botanists never did. Paleontology, Simpson, genetics, Dobrzansky, systematics, of course, Meyer, comparative animal morphology, Wrench, and embryology outside. So one of the things that were excluded was development, and the way that development can fit into a view of evolution, taking into consideration the many, many phenomena, developmental phenomena that embryologists see as they study, and the way that they try to translate their observation of ontogeny into some generalizations about phylogeny. Now, they were not very naive. They were not doing all kinds of Hegelian kind of generalizations, overgeneralizations, but they were trying really to see what does it mean if you have this particular constraints in development and this particular way things happen during development. How does this knowledge sort of reflect on evolution? And the way that they were thinking about it didn't fit with the modern synthesis as it was described and presented by Dobrzansky, Meyer, Simpson, especially this two. Okay, so one of the people who were talking about, who were trying to do such a synthesis, as I said, was Conrad Waddington in Britain. There was another person, Ivan Schmalgausen, in the USSR. He was also doing this. Schmalgausen had a not very happy fate. He was exiled by Lysenko and didn't continue this kind of work. Waddington was a very famous and very well-known geneticist in Britain, but somehow his effect on evolutionary thinking was not very profound. He was very well known. He had a huge, he had a position in Edinburgh, and he was running the genetics institute there, but somehow. Anyway, he was talking about epigenetics, and for him, epigenetics was the layer that connected the genotype and the phenotype. It was the sum total of the causal relations that bring the phenotype into being, starting with the genotype. This was, and he was trying to understand what was happening at this level. Now, I'm not going into the history of epigenetics because this is a lecture in its own right, so I can't do it. What I want to say is that the concept of epigenetics as defined by Meyer has changed. And today, when we're talking about epigenetics, we're talking about a slightly modified term. This is what happens in all kinds of terms in the history of biology. All the time we're updating and changing things. So today, when we're thinking about epigenetics, we're thinking about the processes that lead to long-term, persistent, and to some extent autocatalytic developmental effects. Autocatalytic because things will not be persistent if they're not autocatalytic. They will just fade away. Right? At the cellular level, these are the processes involved in cell determination and differentiation, and at higher levels of biological organization, epigenetic mechanisms underlie self-sustaining interactions between groups of cells that lead to physiological, morphological, and I have to add behavioral persistence. So we see something that is continuously happening, and we try to understand the mechanisms that lead to this persistence, developmental persistence. What are these mechanisms? So this is what epigenetics is about. And this is a picture called The Five Mothers that Anat drew. And this picture shows us the inputs into both development, which is nothing new, but also into evolution. So when we're thinking about what we are, why are we the way that we are? So we are the way that we are because of our genetics, right? So we have a little mother there at the right-hand side, the one with the red hair, and she's the one that, and the genetic variations that we have, she's giving it to us through the DNA. So there are inputs of DNA. Then there are inputs, epigenetic inputs. because one of the things that make us what we are also things, the way that our chromatin is constructed and what we have in the cytoplasm and all this kind of stuff. It's very important. So, as mammals, also the third mother is very important because what happens in utero is also very important and it can make a difference in developmental things. Early development, what we eat, how we are cared for, all these kind of things are also important. Here is the fourth mother, the one who is suckling the baby and transmitting antibody molecules to it, as well as food preferences and so on. And the fifth mother is everything. It's mother culture, and I hope the males here, excuse me, there are no males here, but not because of, it's not a counter to the first picture, it's just because it's too complicated as it is, but of course males play a very, very important role in all this. So, now, all these things that I say to you are trivial. Every biologist knows it, nothing new. What is new is that all these inputs are not only inputs into development, they're also inputs into heredity. A difference in epigenetics can make a difference can be transmitted to the next generation. Behavioral, all kinds of physiological differences can also be transmitted in a lineage, and so on. And there are many examples for all these things. And of course, cultural things, which are specific to humans, in its symbolical terms. I mean, there is also cultural transmission in animals. There are animal traditions, but they are not transmitted through symbolic means. So if we are wanting to think about not only inputs into development, but also inputs into heredity, we have to extend the DNA perspective. So there is, and I'm going to focus on something that is called epigenetic inheritance, which is a component of epigenetics. It occurs where phenotypic variations that do not stem from variations in DNA base sequence are transmitted to subsequent generations of cells and organisms. And this notion is used in a wide term, in a wide sense, but also in a narrow sense. And I will focus on the narrow sense, because in some ways it is more challenging and more general. It applies all over biology. So again, if this is a picture, so we can have transmission, genetic transmissions through, if we're thinking about sexual organisms, through the germline. We also have gametic transmission, also through the germline, of some cellular epigenetic variations. We can have also soma-to-soma transmission that bypasses the germline, that go through all kinds of mechanisms through development, developmental reconstructions are transmitted. It can go also through behavior, through social learning, and also in our case through symbolic evolution and through culture. I'm going to talk about this, about the most general, the cellular epigenetic inheritance. So, even when we're talking about cellular epigenetic inheritance, what we have learned in recent years, and again, this is impossible to cover here, even as in an overview, because there is a huge amount of knowledge. And I want to bring you a little bit up to date with regard to certain aspects of cellular epigenetic inheritance. So, there are different ways, different mechanisms that are involved in transmission of epigenetic variations, for example, variations of, not of DNA, but of things that sit on the DNA. All kinds of aspects of chromatin that can be transmitted from one generation to the next. So, and this is chromatin marks can be transmitted. We have learned recently about an RNA, about the transmission of small RNAs, and there is very, very surprising and very important work on these mechanisms today, in C. elegans, but also in plants and in other organisms. There is also the transmission of things like prions, reconstruction of three-dimensional structures, where variations in complexes in the conformation of a protein or a group of proteins can be transmitted from one generation to the next, and this is seems to be very important in yeast. And there are self-sustaining metabolic loops that we can see that the loops which have a positive feedback embedded in them can lead to the reconstruction of the same kind of activity pattern of the next generation. So in all these cases, what we can have is organisms that have exactly the same DNA. They have different biology, and they have, if we're looking at lineages, we see that these differences are inherited within lineages, and yet the different lineages have the same DNA, but they have a different epigenetic patterns, and these are inherited and can be selected. And they can be selected because selection experiments have been done and are being done. Now, I want to give a little bit of more substance to these words. Now, here we're looking at it again when we're talking about chromatin marks. Changes to the chromatin. What do we mean? We mean a lot of things because there are many changes to the chromatin. So here we see DNA, and it is wrapped around a histone core. Histones are proteins around which in all eukaryotes the DNA is bound. And what we see here is that there are this, so we have DNA and we have histones. DNA there are cytosines in DNA, and the cytosines, especially those that are next door to guanosines, CGs, can be either methylated or non-methylated. Here we have it. This is methylation. So this is one kind of chromatin mark. Then we have all kinds of modifications to the histones, and these are not only changing the transcriptional potential competence of the region, but they can be also transmitted from one cell generation to the next. We have all kinds of remodeler proteins that create patterns on the DNA, and again, these patterns can also be reconstructed in the next generation of cells and organisms sometimes. And we have small RNAs, which are also a very, very important system, and small RNAs are regulators, very potent regulators of many of gene activity in eukaryotes, and not only do they have this developmental role, but they can also be transmitted from one generation to the next. So we have all these things. We know about this mechanism. There is a lot we don't know about these mechanisms, but we're learning about them all the time, and there are more and more, and our knowledge of what is going on during the processes of mitosis and meiosis is increasing, because these patterns, for example, methylation patterns or small RNA patterns in C. elegans, also the modifications of histones are transmitted through the germline, through meiosis. And we're learning more and more about this. So what do we know? So one of the things that people say is, all right, so you have a little bit of examples of epigenetic inheritance here and there, but not much. It's not very important. It's really rare. Now, for this reason, myself and Galraz, student, we surveyed the literature by 2008 until 2009, and we found, just made a survey, we found 102 cases in 42 species. Now, I'm not going to go into it because in a way, this information is irrelevant now. And the reason it is irrelevant is that there is so much new knowledge that to say, well, we have this case and that case, so we have 102, so what? In DNA, the number of variations is as big as, it's every site of the DNA can have 4 different variants. What is 102 compared to this? Nothing. Of course, true. But we have to start somewhere, right? So we have to start with what we knew about the literature. Now we have new methods, molecular methods, and we can actually see what is going on at the level, for example, of DNA methylation. This is the stuff that we know better, that we can study best. So, I'll go back to this picture. But here is something that was, work that was done on Arabidopsis. Arabidopsis thaliana, the Drosophila of the plants. Yes, it's the model organism of the plants. And what the people here were doing, they took a line, an inbred line, highly inbred line, that they were growing for 30 generations. Now they had also the seeds from the 3rd generation because they stored them, they froze them. So now this is a highly inbred line. The level of DNA variation is really very, very minimal, hardly any. There is a little bit, there's always a little, but very, very little. And what they looked at was the variation in methylation in DNA methylation, because this is easy to do. They have the techniques to do. And what they found is, we examined spontaneously occurring variation in DNA methylation in Arabidopsis thaliana plants propagated by single seed descent for 30 generations. 114 to 187 CG single methylation polymorphisms and 2,485 CG differently methylated regions, DMRs, were identified, both of which show pattern of divergence compared to the ancestral line. Okay? So we're now talking not about 102 cases. By the way, the 102 cases that I mentioned, there were sometimes many loci that were involved. It was just the studies that I was mentioning. But we're talking now about this level of epigenetic variation. And they say, thus, transgenerational epigenetic variation in DNA methylation may generate new allelic states that alter transcription, providing a mechanism for phenotypic diversity in the absence of genetic mutation. This is then in Science, 2011. There is another paper in Nature. There is a paper in Maze showing something very similar in inbred lines, and so on and so forth. So, now this is not just plants. We have things, we have less, we are beginning to have more information about animals, but there is not as much information about animals as about plants. There is, but not as much. So here is just a very famous experiment where epigenetic inheritance induced what we saw there, by the way, in the Arabidopsis case. What I showed you is spontaneous epigenetic inheritance. It was not environmentally induced, it was spontaneous. We know, by the way, that if we put plants into stress conditions, there is a lot of variation is appearing very, very rapidly. Here we have a case of rats. It's a famous experiment where another rat was given an injection of vinclozolin. She was pregnant, and her litter was exposed. Vinclozolin is an androgen suppressor. Her offspring were in utero were exposed to this nasty toxin. Vinclozolin is something that we spray wine with, by the way. You have to be a little bit careful. And so her offspring had all kinds of abnormalities. We're looking at the male offspring and at the transmission through the germline of the males. It's much easier to look at males because they don't have a lot of cytoplasmic transmission through the cytoplasm and the effects of the uterus. So we can be sure. So if we see enough generations where something that happened is going through males, we can be quite sure that it is going through chromatin, germline chromatin. So what we have here is we have these males that they have all kinds of problems and they are affected. We mate them with normal females, look at their offspring, look at their offspring, look at their offspring, 4 generations, and we see all the time the same phenotypic problems that are induced by the vinclozolin. Moreover, we also see massive changes in methylation in thousands of genes that are also in correlation. We don't know the causal path, but we know that there is a correlation with the phenotypic changes. So these are 2 examples that I just gave you. This is an example, beautiful example, because one of the things, again, people will say, okay, well, okay, so you have a lot of variation. What about natural populations? Maybe it's an artifact of lab experimentation. Maybe in natural populations we don't have that much. It's not very important. Right? You have to answer these things. It doesn't make a lot of sense, but it's of course a very valid question. It's something we must know about. So people especially in the last, of course it started before, but especially in the last 5 years, people are looking a lot at natural populations, trying to see how much epigenetic variations we have in natural populations and whether or not we can say with a certain degree of sense that this epigenetic variation is related to the local adaptations of these populations. It's very difficult. The reason it's difficult is that you always have also genetic variations in natural populations. You cannot neutralize them. Even in the lab it's very difficult to neutralize. But you can do what you can, and in some cases you can combine work that is looking at populations as well as work that you are doing in the lab on individuals from the natural populations. You can, for example, take individuals from two different populations of the same species and make inbred lines out of them, and then look at what is happening in these inbred lines in the lab and see whether or not this is correlated with the kind of thing that you observe at the population level in the field. It's not easy, but one can do it. So here is the monkey flower, Limulus guttatus, and in a study, again, by Scoville and his colleagues, what they showed was they looked at two populations, and in one population was grazed by animals, the flower was grazed by animals, and it had trichomes. You see this little outgrowths, and at the bottom picture, and the other one was not grazed, and it didn't have these trichomes. And we know quite a lot about the genetics of trichomes, so they looked to see whether there are genetic differences. There were no genetic differences. What they found were epigenetic differences at the level of DNA methylation. And when they did the experiments with inbred lines on these populations, this variation was seen to go through meiosis. So we have a reason to say that this is probably epigenetic inheritance. It was involved in this adaptation. It's an obvious adaptation to grazing, and it's one of the few examples that we have from natural populations of an adaptive change which is probably, very likely, epigenetic. There are not many examples, but you have to remember that there were not many examples in the beginning, in the days of genetics. Most of the examples of Mendelian genetics, for a very long time, were all kinds of very strange mutants in Drosophila, which were all deleterious. So here is another species. This is Viola casovlensis, and again there are populations of this flower in all kinds of places in Spain, and they were looking at different populations that were living in different ecological conditions, and they were trying again to see what is the epigenetic variation, what is the genetic variation. In all the cases that when you look at populations, especially of plants, but also of mammals and other creatures that have methylation, not everybody has methylation, you see that there is a huge amount of DNA methylation, in natural population. Very often, much, much greater than genetic variation. This doesn't mean that it's not related, not correlated with the genetics, but it's a fact of life that we have to sort of start to think about. So they did very complicated statistical tests on these populations, and the conclusion was, because these populations also had genetic variations, and they said, well, it's a very complicated case where we seem to have both genetic inputs and epigenetic inputs and correlations between the two which can explain the ecological adaptations of these populations. But what is important from my point of view, I don't want pure epigenetic variations. It's not what I want is that epigenetic variation will be taken into consideration as an important input into heredity, into evolution, into development, into all these things. It's not that I think that epigenetics is solving all the problems of the world and we can throw genetics out of the window. Of course we can't. It's very, very important. So this is what this work showed. And this is another study also from recently on mangroves. Now here we have two populations of mangroves which are very, very similar genetically. This is a 1.5 meter mangrove, this is a 10 meter mangrove, work done in South America and in the Rio de Janeiro state. And they're looking at the genetic variation and epigenetic variation. There is okay. Yes. Yeah, so this is the big mangrove, this is the little mangrove. The two things are very, very similar genetically, very dissimilar epidemiologically, extremely dissimilar. And there are many studies like this now. More and more and more people are going into the field and having a look. Now, I want to say a few words now. So you can say to me, okay, interesting. So there is genetic variation, there is epigenetic variation. Epigenetic variation, but so far, and some of the epigenetic variation is adaptive. Well, it's bound to be, you know. If there is a lot of variation, some of it will be adaptive. Why not? Some of it will be deleterious, some of it will be neutral, and some of it, maybe a little bit, will also be adaptive. But what is new conceptually in this? So you increase the range of variation about which we can talk. Big deal. So, okay, well, it is a big deal, but it's not really a very big deal. It's not really, you didn't show us, except maybe in the, you showed us that it can also be developmentally organized, but you didn't show us that it can be really important for adaptive evolution. Conceptually, beyond and above what we know about genetic variations, which can, of course, and do, of course, contribute to adaptive evolution when they happen to be beneficial to the organism in particular environments. Now, there is something very interesting that is going on at present. This is work that was published 2 weeks ago in Cell Reports. Now, I'm talking about this because it's not the only work of its kind. It's already a second set of experiments that are showing something like similar. What they did was they took Drosophila, and Drosophila, if you expose larvae of Drosophila to kanamycin, to antibiotics, they die. It's a toxin for them. Now, if you put a construct, a genetic construct, into the Drosophila that is detoxifying, that has a gene that is detoxifying the antibiotic, that is breaking down the antibiotic, the larva can live. This we know. Now, what they did was this. They made a construct with the gene that is detoxifying, the neogene gene, some gene that is doing the detoxification, but they put it under the control of a promoter which is tissue specific. Now, in order for the construct to work, for the detoxification to happen, it has to be expressed in the gut, because this is what they eat, and it has to be broken down there. So, and the promoters that they took are not specifically expressed in the whole gut, or in the gut at all. They are expressed all over. So if we put this gene under the control of a tissue-specific promoter, unless this tissue-specific promoter changes its mode of regulation, the larvae will die. And the question is, what will happen? So, if we're looking at this, I mean, the natural answer is, well, of course they will die. They can't live. But they had a reason to suspect that this will not be the case. So, they did this experiment with 9 different tissue-specific promoters, and they found, in 8 of them, that after a lag period, the larvae stopped growing. And after a lag period, they started to grow again. And when they looked at the expression pattern of this gene, they found that it's expressed in a new way. Now, in all of the cases, the number of the frequency of the larvae that survived was very high. In one case, the hairy promoter, it was 100%. So we cannot argue that this is a story of selection. In other cases, it is 20%, 50%, 60%. Also very difficult to argue that it's a selection. And it's occurring in many flies at the same time, in the same way. Another thing that is happening is that you have new phenotypes appearing, popping out. And there is some correlation between the kind of little variant that you see and the kind of promoter that you use. Now, I will not go into the details, but this is the basic experiment, this is the construct that they do, and what we see here is that in order to have high survival, you have to have this pattern of activity. You don't have this pattern of activity initially, but you leave them be, and the organism, you know, if you challenge an organism is not just sitting there, and it's not passive, it's trying to survive. Now, how is it trying to survive? If it can behave, it behaves, and solves it with behavior, but it's also doing everything it can in terms of its biochemistry. in order to survive. It's trying out biochemical networks, if you want. It's like a learning process at the developmental level. And this is what they found, that they have this difference in the way, in the patterns. So here are the actual pattern that they seek with different promoters. This is the pattern that they end up with, right? Now it is and now comes the crunch point. Okay, this is very interesting in its own right, but this stuff is inherited. How is it inherited? They don't really know all the details, but they know that what they're doing, what is happening is that the expression of genes which are belonging to the polycomb family, which are suppressors of chromatin, are suppressors of expression, the suppressors are suppressed. So what they're showing is that there is a change at the level of chromatin. This is not what is inherited. It is just allowing new patterns to emerge. What is exactly inherited in each of the cases, they don't know yet. There is a lot of work already in this, but they don't know all the details. Now, how long is it inherited? Is the other question that we want to ask. And the answer is it depends. In one case, it's 1 generation, in one case, it's 5 generations, and in one case, it's 24 generations. So, and this is, by the way, the general result we're seeing. We are seeing that indeed, the patterns, the stability, the fidelity of expression is variable. Now, I want to say a few words about what we do. Now, so people are saying, well, that's very interesting. We have evidence that there is a lot of epigenetic variation. Some of it is inherited. Maybe there are also strategies which use epigenetic mechanisms that are using to adapt the variation. But we don't really know what is going on. And that was one of the things that happened in the modern synthesis that was very important, was that people started to make models. Models of what happens in populations. Without models of what happens in populations, we can't really, we only have intuitions. We have to have this kind of models because we're not very good at reiterative kind of statistical thinking. We need to have these models. They are really very good help. And there were very few models until the last three years. There were, yes, of course, there were always people who were doing stuff, but there were few. So first of all, what do we have to look at when we're thinking about it? Let's say, take a pure line, where you don't have genetic variations at all, like the inbred lines in Arabidopsis. What are the factors that you have to take into consideration in addition to those that you take into consideration when you think about genetics? So you have the genetic thing, the selection, drift, migration, and expressivity, all things that you have to think about when you're doing models in population genetics. But there are also other things that are specific to the epigenetic system. First of all is the inducibility of the variation. How easily is it inducible? What is the cost of induction? What is its extent? How many individuals are likely to be affected at the same time? And how many regions in the genome are likely to be affected? The dependence on the inducer's presence. How many generations do we need to expose the creature to the inducing conditions? Is 1 generation enough? 2, 3, 4? We have good reasons to think that there are cumulative processes with regards to epigenetic inheritance. There are experiments. There's paramutability, and that is something that is like gene conversion, but at the epigenetic level. And when you have a heterozygote, you can have one heterozygote with one chromatin state, one locus with one chromatin state, the other locus with a different chromatin state, and one chromatin state imposes its own structure on the other. And this is paramutability, so we have to take this into consideration and to see how common it is and whether it is driving the change in the frequency of the particular epi-allele in the population. We have to think about locus-specific transmissibility. With DNA, it's much easier because we assume that, transmissibility is, in general, there is certain fidelity of DNA transmission, but this is not necessarily the case. With epigenetic things, it can be locus-specific. So there are many things we have to take into consideration. And there are models that are beginning to appear. Again, I cannot go into these models and describe them, but there is a list here of people who are doing modeling and are beginning to take into consideration these factors. What all the models show, all of them, and these are population models, we're not talking about macroevolution here, we're all the time talking about what happens in populations, within populations. And what all the models show is that the dynamics of populations under different assumptions, that take both age, this is the epigenetic factors, and G, this is the genetic factor under consideration, lead to surprising changes in dynamics. Something that was not supposed to have stable polymorphism suddenly has stable polymorphism, all kinds of things. Evolution happens much more rapidly or much more slowly than we expect, and so on and so forth. Really very different. So we're looking at populations and in these models, and we're seeing that if we're just taking the genetic factors into consideration, we're seeing this dynamics. When we're taking both into consideration, we're seeing very, very different dynamics. So it is important, at least the models show us, that with very conservative assumptions about epigenetics, by the way, we get very different dynamics. Okay? Now the other thing is, and this I will say, I think I don't have much time. Do I? Okay. Okay. So one of the things that are being asked, and they're rightly being asked, how do you tease apart genetic and epigenetic variation? It's very, very difficult, because in normal populations you have both. So how do you go about it? At least can you think about a general way of doing it? Now, we have been breaking our heads over this problem for quite a while, and here is a way of giving an intuition, a way of doing something about it, and giving a rough estimate of the role of epigenetic and epigenetic heritability that can be attributed to epigenetics rather than genetic factors in populations. So what we have here, if we think about the phenotypic variance, VP, it is made out in a very simple model of quantitative genetic model of variance, the genetic variance, the environmental variance, and we add VC, the epigenetic variance. Now, how do we know? Usually, in classical models, we think what has happened is that the VC, the epigenetic variance, was attributed either to the genetics or to the environment. Now we want to see how can we extract the epigenetics from the genetic? How do we do it? So the trick is very simple. The trick is to keep the genetic relatedness the same, but the number of meiosis to change them. So, because why is it the trick? Because in every meiosis there is a chance of resetting the epigenetic marks, of forgetting. So, if you have two individuals that have the same relatedness, but the number of meiosis that each of them went through is different, the bigger the number of meiosis, the less phenotypic similarity will we see. So let's take two examples. Okay? Here we have. Let's look at a parent and an offspring. A parent and an offspring have genetic similarity of roughly 50%, right? They're 50% similar to each other. Okay? Now, also, two full brothers are 50% similar to each other, right? On the average. Correct? Okay. Now, let's look at some trait that the parent has and the trait that the offspring has. We have one meiosis here and one chance for reset. V is the coefficient of reset. And let's also compare offspring 1 and offspring 2. Each of them went through an independent meiosis. They are still 50% similar, but since there are two meiosis, so separating them, so to speak. So we are expecting that there are two independent chances of reset, and the epigenetic relatedness is a function of 1 minus V. V is the forgetting, Yes, so 1 minus v is the memory squared. Okay, there are two independent things. Whereas here, the chance for reset is v. Now, the same we can show that the grandparent and the offspring here and the grandchild here are separated by 2 meiosis, and the coefficient of relatedness is 1/4, 25%. The coefficient of relatedness between uncle and offspring and nephew is also 25%, but that's 3 meiosis. Now, when we take this into consideration and play a little bit with the mathematics, not much, because I'm not a very good mathematician, so I can't do anything very complicated. So we can actually isolate these things, and also this work was done by a very good mathematician, by the way, a student of mine, who is good at this kind of stuff. And so we can isolate this is the memory coefficient, I minus b. We can, and this is VC, and this is VA, and if you want to see how we derive it, you're welcome to the table. But basically, what we're looking at is covariances between, this is half sibs, correlation between uncle and nephew, offspring parent, half sibs, offspring parent, half sibs, and we can give a rough estimate of this, of the epigenetic variance, of the memory coefficient, and also of the additive genetic variance from this. So there are ways of doing it. Nobody did it yet. I mean, people are writing to us about it, and we think that sooner or later something will happen. The problem is that we need information here, for example, about uncles and nephews, which usually doesn't exist in the literature. So people have to measure this kind of thing. It's not a big deal, but the work somebody has to do it. So there are ways of also estimating epigenetic heritability, and we made all kinds of simulations showing that it is likely to be significant. Okay. Now I want to say a few words about macroevolution. We were talking about microevolution, and we were talking about what is happening within populations. Now, what we know about processes of genomic shocks and ecological shocks, and genomic shocks include things like polyploidization, you double the number of chromosomes, or hybridization, often followed by chromosome doubling. These are shocks. You know, you take two genomes that are far away from each other, put them together, and you expect the stuff to work. I mean, it's not likely that it will work very easily, right? And you put, it's a shock, and very often it doesn't. Right? Very often it doesn't. Now, we know a lot about, now we can do all kinds of synthetic hybrids. We can take we can take two species from two populations, put them in the lab, hybridize them, and then put some colchicid or something, or not even colchicid, something, or just wait if we have a lot of space, and some of them will also duplicate, double the chromosome number. This is important for meiosis to work in most cases. Now that's very well known it has been known for ages. What is new, relatively, is that during these processes, like hybridization, polyploidization, and also under various really nasty ecological shocks, very big changes in DNA methylation are happening. But really very big. I mean, if you are looking here at these polyploids of wheat that one can do in the lab, you are seeing a change of 13% of the methylation is changed. It's a big, big change. Wheat has a very big genome. There are many other people who are doing this kind of things and are showing that there are big epigenetic changes happening. There are also genetic changes happening. I want to stress this. These genetic and epigenetic factors, I have been talking about them as if they're separate here for heuristic reasons, because we have to separate the things a little bit. And they are to some extent, and under some, there are sometimes we can separate them. For example, in inbred lines we can do it, and sometimes there is a degree of independence. Usually there isn't. And the epigenetic mechanisms are also related to what James Shapiro calls mechanisms of natural genetic engineering. And he gives he has a very important, I think, new book, Evolution in the 21st century, evolution, a 21st century view. And where he documents all the kind of things that happen in genomes, in the genomes of mainly microorganisms, but not only, which are usually pushed under the carpet by evolutionary biologists. Okay. So we have what we know that is that genomic shocks and ecological shocks are leading to very big changes in the epigenome. And they are leading to a lot of new variation also. So here is a model that was done by Rapp and Wendell, who are working on cotton, cotton hybrids, and are also seeing the same kind of thing that everybody who's working on these hybrids is seeing, and that is that there is a huge amount of epigenetic variation that is appearing as a result of this shock. And what they say here is here we have this, we have a population, and this population has its own variation. And now we take this population through some kind of shock. This can be a genomic shock, it can be an ecological shock, it can be a very extreme inbreeding situation where we take it through a very big bottleneck or something like that, and we look at what happens. And one of the things that what happens is, of course, that the genetic variation is very often becomes very restricted, but the epigenetic variation is jumping up to all directions, and what we have is this new expression phenotypes, which we can then select. We select them, and we select their genetic correlates or anything that stabilizes them at the genetic level, so we can have some kind of genetic assimilation. Right. Now, I don't want to... Now, what I wanted to say in this, what I'm trying to say in this lecture is to say to you, look, it's time for us to take into consideration the new knowledge that is coming from molecular biology, molecular developmental biology, about epigenetics. We have a lot of things we don't know, and I'm listing here very few. For example, people have, and you know, there are things we can do and should do, I think, in order to answer questions. For example, we can look at recent speciation events and try to figure out how big a role is there for epigenetic kind of variation. We can look at domestication, people are beginning to do this, and look at the epigenetic component of domestication, which was pointed out, by the way, very early on by Dmitry Belyaev in the USSR, a great biologist, a great geneticist. There is a very recent study about chicken now, and in this study, what we see is what they are doing is they are comparing the wild ancestor of the chicken and one of the Leghorn, I think, strain of domesticated chicken, and they looked at the epigenetic and the genetic variation. And what they see is they have some genetic changes, of course, but not many. Massive epigenetic changes. So, very interesting questions, and we can look at domestication. We can also do domestication in the lab, or start a domestication process in the lab. And we have this domestication experiment that were done by Belyaev on silver foxes that have been going on for 50 years. 50 years is not a very long time, and it's very interesting to see what is happening there at the epigenetic level. We can do experiments which simulate extreme shocks, both ecological and genomics. What are the effects on the epigenome? People are doing this. Is this kind of phenomena that I was describing to you in Drosophila, that we can call it genomic accommodation, is it something, epigenomic accommodation, is it something more general? I mean, we have seen it in Drosophila. There is also an experiment that showed that this kind of, with similar kind of logic, that it also happens in yeast. Is it a general phenomenon? Can we generalize on the basis of this kind of thing? It's very important to know. How can we model process of macroevolution? We are good at modeling microevolution. It's easy. How to model macroevolution? Taking into consideration this kind of things. So there are lots of things one has to do and one has to think. Now the kind of position that I'm putting forward here is not without, there are a lot of people that are saying really, you know, come on, it's just an extension of what we know. It's not really beyond It's an extension, but it's not something, we don't have to rethink things. We just have to, okay. So here is a quotation. A naive or contrarian interpretation of epigenetic discoveries can raise the spectre of Lamar, a terrible thing, if they allow for the limited inheritance of acquired characteristics, or even that of Lysenko. This is a really nasty guy, but it's always used as a weapon, like Hitler, right? Who believed that environmental manipulations could induce radical and heritable phenotypic change. It is therefore important to stress, first, that many epigenetic responses are determined and controlled by DNA changes, of course true, and second and more fundamentally, it makes no difference to the basic mathematics of evolution by natural selection whether or not the heritable changes involved consist solely of alterations in DNA sequence. A full understanding of epigenome may make important and unexpected contribution to evolutionary development of biology, but this will extend, not overthrow, the Darwinian synthesis. Now, nobody wants to overthrow the Darwinian synthesis. What we need to overthrow is the neo-Darwinian synthesis, not the Darwinian synthesis. Darwin was a great, the insights of Darwin are very important. Also, some of the insights of the modern synthesis are very, very important. But we cannot think in terms of what Bayer said 60 years ago. I'm sorry, 60 years have passed. Something happened in the world. So, I think that they're wrong. I think that there is very good evidence that it does make a hell of a lot of difference to the mathematics of evolution by natural selection. The results are very different. The models are not terribly different, but they're a little bit more complicated. That's all. Not terrible. So I think, to conclude, to put it, that the neo-Darwinian synthesis has to be changed. Not the Darwinian, the neo-Darwinian. Not the Darwinian basic idea of the recognition of selection. On the contrary, I think that the role of selection has to be expanded in some way to incorporate the kind of selection processes that happen within cells. And the Darwinian synthesis has to be extended because one of the things I didn't talk about is that we can have, taking into consideration the systems, we can have very rapid evolution. So not all evolution is gradual. We need an extended notion of heredity that is not only based on variations on DNA, although this is fundamental and very important. We need to have a developmentally informed notion of evolution, and we're beginning to have it because we have evo-devo, and epigenetics is part of evo-devo. And the difference that it makes is, it makes a difference. The epigenetic consideration make a difference to the rate, to the adaptability, and to the specificity. Can make a difference of evolutionary change. Thank you very much.